Watch a goshawk thread the needle through a dense forest canopy at 50 miles an hour. Every millisecond requires hyper-focused action selection and flawless coordination, all executed without a mammalian neocortex. For decades, anatomists looked at the avian brain and saw what they assumed was a primitive structure. The prevailing theory held that the forebrain was basically just a massive, instinct-driven basal ganglia, lacking the sophisticated layering of a human cortex. That theory collapsed once researchers began mapping the brain at the molecular level. By tracking evolutionarily conserved homeobox genes, the transcription factors that regulate how the brain develops in embryos, scientists proved that the avian forebrain is highly compartmentalized and structurally homologous to our own. The key landmark to understanding this architecture is a distinct anatomical boundary line called the pallial-subpallial lamina, or LPS. This lamina cleanly separates the upper region, the dorsal pallium, from the basal subpallium below it. This diagram illustrates the coronal mapping of that subpallium beneath the LPS boundary line. Rather than a singular mass, modern mapping confirms it comprises 6 distinct, highly specialized neural systems. Taking up the bulk of the ventral medial forebrain are the basal ganglia zones. We have the dorsal somatomotor region in red, which handles physical movement, and the ventral visceral limbic region in tan, which regulates affective and motivational states. Tucked closer to the diencephalon are the remaining 4 systems: the extended amygdala in dark blue, the corticopedal system in green, the septal divisions in yellow, and the preaptic system in light blue. Together, this 6-part architecture acts as a direct physiological bridge. It takes complex cognitive decisions generated in the upper pallium and translates them into precise executable commands for the autonomic nervous system. To understand how birds move with such precision, we have to look closely at the dorsal somatomotor basal ganglia. This region operates as the exact functional equivalent to the mammalian caudate putamen. We know this because histological stains reveal the same neurochemical signature. The avian striatum is densely packed with both dopamine and acetylcholine esterase, identical to mammalian motor centers. Look at panel C in this micrograph series. The dark staining you see is tyrosine hydroxylase, an indicator of dopaminergic input. This dense footprint maps the exact borders of the medial and lateral striatum, sitting immediately below the LPS boundary. Movement initiated within these striatal structures doesn't rely on a single on-switch. It operates entirely through a parallel dual pathway circuit. The direct pathway sends excitatory signals from the pallium to the striatum, which inhibits the internal globus pallidus, disinhibiting the thalamus to trigger movement. Concurrently, the indirect pathway routes inhibitory signals to the external globus pallidus, suppressing the thalamus to block unwanted movement. A bird navigating a forest canopy requires both pathways to fire simultaneously. The direct circuit executes the precise wing tuck, while the indirect circuit aggressively suppresses any conflicting muscle twitches that would throw the bird off balance. This deeply conserved pull-pull logic proves that advanced motor selection does not require a mammalian cortex. Tetrapods solved this mechanical problem millions of years ago, deep within the subpallium. But those striatal pathways are essentially an engine waiting for fuel. To actually initiate action, they require constant modulatory drive from deep within the brainstem. These histological stains map the primary dopaminergic engines feeding the basal ganglia, the ventral tegmental area, and the substantia nigra pars compacta. Specifically, their A10 and A9 cell groups project the dense dopamine required to regulate voluntary movement and reward processing. The system also needs a distinct chemical trigger for acute arousal and stress. This comes from dense noradrenergic inputs. Down in panel G, the A6 cell group in the locus coeruleus pumps noradrenephrine directly into subpallial regions like the nucleus accumbens. Without these specific brainstem nuclei supplying the chemical context, whether that is the reward of finding food or the sheer panic of an approaching predator, the subpallium has no biological motivation to bias one action over another. Once we understand how movement is driven, we have to look at how affective states are processed. That happens in the ventral visceral limbic basal ganglia, the subpallial territory dedicated to translating motivations into behavior. Look at the intense substance P immunoreactivity across these 4 transverse sections. The heavy staining maps the visceral limbic pallidal subdivisions. specifically the ventral pallidum and the tuberculum olfactorium. Sitting at the apex of this visceral limbic network is the nucleus accumbens. It functions as the ultimate limbic motor integrator, catching diverse inputs from across the brain. This comparative atlas shows the avian accumbens is partitioned into distinct subterritories: the core, shell, and rostral pole. Notice the chicken brain on the left compared to the pigeon on the right. The pigeon's accumbens extends much further rostrally, highlighting localized evolutionary tuning. These partitions have vastly different jobs. The accumbens core wires directly into the overlying somatomotor pathways. It is the region responsible for the instrumental learning of brand new motor skills. The accumbens shell, however, is wired for visceral integration. It receives inputs regarding internal drives and lights up specifically in response to stressful or highly reinforcing stimuli. By splitting the nucleus accumbens into these specialized zones, the bird possesses a dedicated interface to translate raw, abstract motivational signals straight into executable behaviors. For those behaviors to actually alter the bird's heart rate or digestion, the subpallium needs a direct pipeline to the lower brainstem. That pipeline is the extended amygdaloid complex. Panel A highlights the central extended amygdala in green. Notice how it forms a continuous sublenticular pathway running directly beneath the globus pallidus, projecting into the lateral hypothalamus and parabrachial nucleus. By linking to these deep autonomic centers, it regulates ingestion, fear, and stress. Moving to panel B, we find a distinct second pathway, the medial extended amygdala and its bed nuclei. Instead of pure motor or fear inputs, the medial extended amygdala receives dense projections directly from the olfactory bulb and vomer nasal systems, processing vital chemical cues from the environment. This medial pathway is also heavily concentrated with gonadal steroid receptors, making it highly sexually dimorphic. It takes those chemical and hormonal inputs and coordinates immediate reproductive and defensive autonomic responses. These extended amygdaloid corridors serve as the brain's translation engine. They ensure that external environmental threats and internal hormonal states are instantly converted into hard, measurable autonomic output. When we zoom out and look at these distinct regional systems together, the avian subpallium reveals itself as a highly efficient, top-down computational pipeline. Excitatory decisions originate up in the pallium and cascade down into these striatal and amygdalar processing hubs. Inside those hubs, the signals are rigorously parsed by the basal ganglia's push-pull logic, while simultaneously being throttled and modulated by brainstem dopamine and norepinephrine. The finalized action plan is then fired through the extended amygdala, transmitting a clean, unhesitating set of instructions directly into the autonomic nervous system. The underlying mechanics here are elegant. The avian brain manages visceral reactions, affective states, and reproductive timing using the exact same precise inhibitory-excitatory logic that it uses to move its wings. The old assumption was that sophisticated cognition and autonomic control were locked behind the evolution of a mammalian cortex. The anatomy proves otherwise. Convergent architectures across tetrapods ensure that rapid, highly filtered action selection is a universal vertebrate trait. When a massive flock of birds turns as one, they are executing the real-time calculations of this integrated subpallial network, flawlessly navigating the demands of the natural world.